My name is Caitlin, and I am a research associate within Core Services located in the Department of Chemistry at the University of Alberta. Today I will be talking about the biosynthesis of carbohydrates. There are two main types of carbohydrate active enzymes, Glycosyltransferases and glycoside hydrolases or Glycosidases. Today we'll talk about the mechanism of each enzyme and how we can utilize Glycosyltransferases and Glycosidases in the synthesis of oligosaccharides. We will then briefly talk about how we can use these enzymes to synthesize analogs of oligosaccharides by using unnatural substrates. Glycosyltransferases catalyze the formation of glycosidic bonds. Each coupling requires a glycosyl acceptor and a glycosyl donor. The donor species is always a sugar nucleotide or a glycosyl phospholipid. Glycosyltransferases are classified as either Lenoir or non-Lenoir based on the type of glycosyl donor that they recognize. The Lenoir transferases recognize sugar nucleotides, whereas the non-Lenoir transferases recognize sugar phospholipids. They were named after the 1970 Nobel Prize-winning chemist Louis Lenoir, who discovered sugar nucleotides. The glycosyltransferases involved in the assembly of mammalian oligosaccharides recognize 1 of 10 activated sugar donors. 9 are sugar nucleotides and 1 is a glycosyl phospholipid, as illustrated below. It's important to note that bacterial and fungal organisms have a much broader range of carbohydrate residues in their glycoconjugates, and therefore they have a larger number of glycosyltransferases and activated sugar donors, both sugar nucleotides and glycosyl phospholipids. There are two pathways in which glycosyl donors are synthesized. The Lenoir pathway and the Salvage pathway. In the Lenoir pathway, all sugar nucleotides are derived from glucose, as shown here. In the next few slides, we will take a closer look at the synthesis of some of these key intermediates, but for extra information on the synthesis of these intermediates, please see the paper indicated in the left corner. In the biosynthesis of the UDP derivatives of glucose, galactose, xylose, and glucuronic acid, glucose is first phosphorylated to form the glucose 6-phosphate, which is then converted to glucose 1-phosphate in the presence of a phosphoglucomutase. Glucose 1-phosphate is then converted to the UDP-glucopyranose through the use of a phosphorylase enzyme in UTP. From the UDP-glucopyranose, UDP-galactopyranose is formed through the use of an epimerase enzyme. The UDP-glucopyranose can also be converted to UDP-glucuronic acid through the reaction with a dehydrogenase enzyme, and a subsequent decarboxylase will convert the UDP-glucuronic acid to the UDP-Xylopyranose. The UDP derivatives of N-acetylglucosamine and N-acetylgalactosamine stem from the intermediate glucose 6-phosphate. Glucose 6-phosphate is converted to 2-keto-glucose 6-phosphate, which is transformed to glucosamine 6-phosphate. Upon acetylation of the nitrogen, the phosphate group is transferred to position 1, which is then converted to the UDP sugar. The UDP glucosamine is converted to the UDP galactosamine in the same process that the UDP glucose was converted to the UDP galactose. From the 2-keto-glucose 6-phosphate intermediate, mannose 6-phosphate is formed. The phosphate group is transferred to position 1, which undergoes a reaction with GTP to give the activated sugar GDP-mannopyranose. From the activated sugar GDP-mannopyranose, GDP-fucopyranose is formed, as well as the dolichophosphomannose. The second pathway in which glycosyl donors are synthesized biologically is through the salvage pathway. The salvage pathway procures monosaccharides from the degradation of glycans within cells. These monosaccharides are phosphorylated at position 1 and then reacted with UTP to form the activated UDP sugar donor. A number of crystal structures of glycosyltransferases have been solved since the early 2000s, and they have shed light on different types of mechanisms that may take place in the active site. Glycosyltransferases are classified as either retaining or inverting, and the designation describes what happens to the stereochemistry at the anomeric position of the glycosyl donor during the transfer. In the retaining mechanism, it was first proposed that a nucleophilic residue on the enzyme attacks the leaving group, forming a covalent bond between the enzyme and the glycosyl donor. The glycosyl acceptor then displaces the enzyme in a double SN2-like reaction that results in the net retention of stereochemistry at the anomeric position of the glycosyl donor. Clausen and coworkers have presented some mass spectrometry evidence for the presence of the covalent bond between the enzyme and glycosyl donor. However, there has been emerging evidence that indicates an SNI or a substitution nucleophilic internal-like mechanism is a more accurate picture for the mechanism of a retaining glycosyltransferase, where there is a front-facing leaving group. In this mechanism, there is direct attack of the glycosyl acceptor on the glycosyl donor, and the transition state is expected to be highly dissociative with a significant oxocarbenium ion characteristic. The inverting mechanism involves a basic residue on the enzyme which deprotonates the glycosyl acceptor, allowing for nucleophilic attack on the glycosyl donor in an SN2 reaction that results in the inversion of stereochemistry at the glycosyl donor. Using glycosyltransferases to synthesize oligosaccharides can be useful because they offer some advantages over conventional chemical synthesis. In glycosyltransferases, there are typically two acidic residues in the active site, and they have higher substrate specificity than glycosidases. The reactions are regio and stereospecific, and they require no protecting groups. Many of the reactions can be run in water. However, there are some drawbacks, such as the enzymes themselves can be difficult to express, purify, and store, as well as the enzymatic reactions are inhibited by the nucleoside diphosphate byproduct produced in the transferase reaction. Despite these drawbacks, many transferases have been widely studied and are employed in the commercial synthesis of some oligosaccharides. Glycosyltransferases are typically used in conjunction with other enzymes. Sugar nucleotides are expensive to buy, so they are typically synthesized in situ by using a phosphorylase in conjunction with the glycotransferase. To degrade the nucleotide diphosphate, which inhibits the transferase reaction, pyruvate kinase is used to regenerate the UTP needed for further catalytic cycles. Multi-enzyme reactions alleviate the pressures and high cost of the starting sugar nucleotide and drive the enzymatic reactions to completion by removing byproducts that inhibit these reactions. Glycosidases, or glycosyl hydrolases, are classified as either exoglycosidases or endoglycosidases. The exoglycosidases cleave only the terminal glycosidic bonds, whereas the endoglycosidases can cleave the internal glycosidic bonds. Glycosidases can be further characterized as retaining or inverting enzymes, which again describes the stereochemistry at the anomeric position of the cleaved glycoside. The glycosidase enzyme can contain two acid-base residues within the active site. The basic residue will attack the anomeric position, which releases the glycosyl group and can be protonated by the acidic group residue in the enzyme. When water is present, it will attack the covalent bond between the enzyme and sugar in an overall double SN2 mechanism, which results in the retention of stereochemistry at the anomeric position. In the inverting mechanism, the protonation of the glycosidic bond is the first step by the acidic residue on the enzyme active site. Water displaces the protonated glycosyl group, resulting in an inversion of stereochemistry at the anomeric position. It is more difficult to use glycosyl hydrolases in the synthesis of oligosaccharides. The enzymes themselves contain two acid-base residues in the active site, and they have very high substrate specificity. The natural equilibrium of this reaction favors the hydrolysis of the glycosidic bond, which means in order to utilize these enzymes in a synthetic reaction, you must reverse the equilibrium. For this reason, you cannot run the reactions in aqueous conditions, and you must use anhydrous conditions. Reactions that use these enzymes typically suffer from low product yields and mixtures of products. For these reasons, glycosidases are typically not used in large-scale synthesis. Mutated glycosidases can be useful in synthetic reactions. One of the two acid-base residues in the active site of the glycosidase enzyme is mutated to a non-nucleophilic residue, and typically a synthetic glycosyl donor, such as a glycosyl fluoride, is used due to having a small mobile leaving group. The reaction is no longer in equilibrium, which means the reactions can be run in aqueous conditions. Additionally, sometimes a p-nitrophenyl glycoside is used because they bind strongly to the aglycone site but are not cleaved by the enzyme. The rigid substrate specificity has generally inhibited attempts to use unnatural substrates in glycosyltransferase reactions. This makes synthesizing analogs of oligosaccharides difficult. Many enzymes that recognize unnatural substrates suffer from very slow reaction rates. However, an example of successful integration of unnatural substrates is in the use of sialotransferases. It has been found that sialotransferases can tolerate many different substitutions on the nitrogen at position 4, and researchers have shown that using unnatural analogs of MANNAC result in unnatural sialic acid analogs. Because of the propensity of sialo sugars to be expressed on cell surfaces, unnatural MANNAC derivatives have been created to provide handles for further chemical modification. Common modifications are the levulinal and azidyl functionalities, allowing for cell surface labeling with chemistry that is orthogonal to other cell surface functionalities. Today we have talked about glycosyltransferases and glycosyl-hydrolases, the two main types of carbohydrate active enzymes. We have briefly discussed how transferases and hydrolases can be used to synthesize oligosaccharides, and we touched on how unnatural substrates can be incorporated into the enzymatic oligosaccharide synthesis. If you would like to learn more about the interactions between various proteins and carbohydrates, please refer to our module entitled Carbohydrate-Protein Interactions.